Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes. At Informationboxticket Lifestyles, we often explore the unusual, intricate, and sometimes downright bizarre journeys of scientific breakthroughs and teamwork. Yet, one of the most extraordinary tales, surprisingly, is one we have barely touched on, and it's a major one. The long and challenging journey over several decades to fully understand HIV and AIDS and to discover effective methods for their prevention and treatment. Since the onset of AIDS epidemic, around 88.4 million individuals have contracted HIV, with approximately 42.3 million losing their lives to the disease. Each of these figures is staggering on its own, but together they reveal the immense toll of a disease that has caused profound loss, yet also highlight how, with access to proper treatment, HIV is no longer a guaranteed death sentence. In this video, we are going to share that story with you. There's a lot to unpack, so we'll break it into two parts. In this first part, we'll explore how scientists identify HIV, how the infection progresses into AIDS, and the origin of virus as we today understood them. In the next video, we'll dive into the early treatment efforts, the breakthrough introduction of antiretroviral drugs, and the revolutionized care and the innovative strategies scientists are now exploring for prevention and maybe even a cure. But before we get into all that, let's start with the basics. HIV, or human immunodeficiency virus, is a type of retrovirus that targets the immune system, specifically CD4 T cells. Being a retrovirus means it carries its genetic code in RNA, which is less stable and more flexible than DNA. After infecting a cell, HIV uses a unique enzyme to convert its RNA into DNA, which it then integrates into the host genome, essentially hijacking the cell's machinery. If that sounds sneaky, that's because it is. This ability to hide within the host gene is one of the key reasons HIV has been so challenging to treat, a topic we'll explore more deeply in the next video. CD4 T cells, the one targeted and eventually destroyed by HIV, are a type of white blood cells known as helper T cells. Their job is crucial. When they detect a threat, they release signaling proteins that help rally and coordinate various parts of the immune system. In short, they are essential for a strong defense immune. You definitely want them on your side. HIV is transmitted through bodily fluids such as blood, semen, vaginal fluid, and breast milk. That's why it can spread through sexual contact, shared needle, breastfeeding, and any other exchange of these fluids, with one major exception. Saliva isn't on that list. Saliva contains various substances that stop HIV from being infectious, including antibodies and several antimicrobial proteins. So unless there's a significant amount of blood mixed with saliva, it can transmit the virus. When someone is first infected, they may experience flu-like symptoms such as fever, headache, rash, sore throat, and muscle or joint pain. Because the virus is rapidly infecting cells, the immune system is reacting to fighting it off.
If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated.
But after a few weeks, those symptoms subside because the body begins producing specific antibodies that help control the virus and prevent it from spreading unchecked. From there, most people feel fine for a long time, sometimes even for decades. That is until the virus has gradually destroyed too many T cells, weakening the immune system to the point where it can no longer defend against pathogens, anything harmful or infectious. That's when a person is considered to have AIDS or acquired immune deficiency syndromes.
AIDS is typically diagnosed when a person's T cells count drops below 200 cells per microliter of blood, far below the normal range of 500 to 1500, or when they develop what's known as an opportunistic infection. These are infections that a person with a healthy immune system would typically fight off without much trouble. But for someone with AIDS whose T cells have been wiped out by HIV, those same infections can become life threatening. In fact, it's often an opportunistic infection that ends up being fatal. Now, some of that might already be familiar, but imagine, just for a moment, that you have never heard of HIV or any of that what we have just talked about. Because in 1980s, none of these basic facts were known. All doctors could see was that otherwise healthy young gay man was suddenly coming down with extremely rare infection and cancers, and those illnesses were proving fatal. An immunologist at UCLA was among the first to recognize the emerging pattern. Between late 1980s and the spring of 1981, he encountered 5 patients, all gay men in their 20s or 30s, suffering from a rare and alarming form of pneumonia. A fungus had taken hold inside their lungs. Normally, this type of fungus wouldn't pose any threat and certainly wouldn't infect the lungs. But in these men, it had, and it was severely affecting their ability to breathe. On top of that, they all showed signs of oral thrush, essentially yeast infection in the mouth, and had alarmingly low count of CD4 T cells. By June, when the immunologist documented his findings for the CDC's weekly morbidity and mortality report, 2 of the 5 patients had already died. Just a month later, a dermatologist in New York submitted a similarly alarming report, this time involving Kaposi's sarcoma, a rare cancer marked by blotchy purple lesion on the skin. Within just 2.5 years, 26 gay men in New York and Los Angeles had been diagnosed with Kaposi's sarcoma. Some also suffered from unusual fungal pneumonia, and 8 of them had died. It's difficult to imagine now, but at that time, scientists had no clue what was causing the illness. They didn't know whether it was a toxin, an infectious agent, or something else entirely. And if it was an infection, they had no idea how it was being transmitted, leaving them unable to advise the public on how to stay safe. The connection to gay men was definitely noticeable. And in the early days, many referred to the mystery illness as GRID, short for gay-related immune deficiency. It was often called the gay cancer or even the gay plague by the public. But the disease wasn't confined to gay men. It was also appearing in hemophilics, people whose blood doesn't clot properly. Hemophilic receiving clotting factor derived from the donated blood, which put them at risk. Doctors were also identifying cases in HIV drug users, women, infants, and heterosexual men. Notably, 20 recent immigrants from Haiti had fallen ill, and none of them reported being gay. Those clues were crucial because they pointed scientists towards the conclusion that this disease, now officially named as AIDS, was likely infectious and probably spreading through blood. Other diseases like hepatitis B were known to transmit in similar ways. So in March of 1983, the CDC released a warning urging doctors to take precautions with blood transfusion and noting that the disease appeared to be transmitted through both gay and heterosexual sex. Debates about the details like whether saliva could transmit the disease would come later. For now, the urgent question was, what exactly was the infectious agent? Scientists were in a race to identify the cause of the illness. French molecular biologist Luc Montagnier suspected a virus since the blood product used by hemophilics were already filtered to remove bacteria and fungus, but viruses, being much smaller, could slip through those filters. Together with his colleague Francois Barnes-Heroisis, Luc Montagnier examined cells from AIDS patient and discovered a retrovirus. At about the same time, Robert Gallo from the NIH in the US also identified a retrovirus in samples taken from the AIDS patients. Both research groups published their findings in May 1983, and soon another team discovered yet another retrovirus. Each of the viruses was initially given a different name, and at first it wasn't completely clear that they were the same virus. But they were, and by 1986, the cause of AIDS was officially named as HIV. So HIV was the culprit, but where had it come from, and why had the epidemic emerged in 1980s, a decade known for big hair, Michael Jackson? While some researchers were racing to identify what had made AIDS infectious, others observed that macaque monkeys seemed to suffer from an AIDS-like disease. In 1985, one group decided to collect blood samples from these sick monkeys, and they discovered a virus similar to HIV. This virus was later named as SIV, which stands for Simian Immune Deficiency Virus. Researchers began to suspect that HIV might have originated from our primate relatives crossing the species barrier. After extensive research, they determined that the virus responsible for epidemic was closely related to the chimpanzee version of SIV, and it was these primates that had passed it to humans. But how exactly did this happen? While there's no easy way to phrase this, some scientists agree that the reason SIV made the jump to humans, what's known as spillover, was due to our consumption of bushmeat or wild game, specifically monkeys and chimps. This is referred to as the cut hunter hypothesis. It suggests that while butchering a chimpanzee, SIV-infected chimpanzee's blood could enter a small cut on the hunter's hand or possibly splattered into the mouth. The virus is close enough to human biology to infect the hunter, and over time, if the hunter spreads the virus to enough people, it evolves into HIV we recognize today. Spillovers like this occurred many times. Scientists have determined this because the virus mutates rapidly, and by examining genetic differences, they can trace multiple lineages of the virus, each linked to a separate spillover event. We have traced the current epidemic back to just one of these lineages, called M for male. By studying chimpanzees' urine and feces, researchers believe that the chimps that passed this version of the virus to humans lived in southwestern Cameroon, in the forest near the Congo. And based on the oldest known blood samples that now contain HIV, which date back to 1959 and 1960, scientists estimate that HIV-1 first infected humans around 1908. If that feels like a long time, remember it takes time for a virus to spread. By 1920s, it believed that the virus traveled downriver, carried by a person, of course, to the growing city of Kinshasa, which was then known as the Belgian colonial city of Leopold-Diabelli. There were few women in the area except for prostitutes, so experts believe HIV spread through this route and possibly also through injectable drugs used by colonists to treat certain tropical and venereal diseases. This was before disposable syringes, and nurses were attempting to treat many patients with only a few syringes. So they may have simply rinsed them with alcohol before using them on the next person. So the very methods intended to prevent the spread of disease may have actually been contributing it. Over time, infected individuals in Kinshasa moved to other places and they carried the virus with them. Due to the virus' rapid mutation, we can categorize the viruses into 9 distinct subtypes, which helps us trace how HIV spread from Central Africa to other parts of the world. Several subtypes spread across different regions of Africa. Subtype C moved southwards and eventually reached India. Subtype B traveled to Haiti, and through a series of historical events, eventually it made its way to United States. In 1960s, when the Belgians left the Congo, French-speaking Haitians began arriving to work as doctors, lawyers, and other professionals. However, with the formation of Zaire in 1965, the immigrants felt unwelcome and returned to Haiti, bringing HIV along with them. In Haiti, HIV spread rapidly, possibly due to plasmapheresis center where people were paid to donate their blood plasma. The machine used at the center mixed the blood of different donors, facilitating the transfer of viruses. By 1982, nearly 8% of a group of young mothers in Port-au-Prince slum tested HIV positive, an alarming high figure. HIV is believed to have entered the United States around 1969, likely through one infected individual or a unit of plasma from Haiti. It took about a decade for anyone to notice, but by then it was too late. The epidemic had begun, and HIV was not only in Americans but also in Europe and Asia. And now that it was there, we needed to figure out how to fight it. But we'll get to that in the next episode of this mini-series. In meantime, thanks for watching this episode of Informationboxticket Lifestyles, and if you want to learn more about HIV and all kinds of other science, you can visit my YouTube channel and subscribe.